All right, welcome everyone. We know that people will be coming in over the next few minutes and will be entering from the waiting room. We just wanted to have a disclosure for all of our participants to please be advised that this event will be recorded and digital screenshot may be taken for purposes of documentation. We love pictures, even if they're on Zoom. But if you do not want to have your name or face potentially in a photo, please kindly turn off your camera or change your name to maybe your other first name or initials if you want. Please also have any phones or devices readily available to scan QR codes near the end of the summit for both evaluation, which will be very important, as well as our digital virtual swag bag that has a lot of linkages to upcoming events, as well as really wonderful resources. We appreciate all of our partner organizations on our planning committee for this third annual summit.
So welcome everyone. I'm so pleased to have you join us today for this third annual Chicago Gynecological Cancer Researcher Survivor Summit. My partner in crime here, Dr. Anita Lee, as well as our planning committee, have put on a great event for you today. And we just appreciate you all. You could have done a lot of things with this Saturday, but you're here with us. So we definitely appreciate that.
We are so excited to have this event continue, and we really want you to use this as a branching point for how you think of yourself engaging with GYN cancer research, whether it be as a patient, a caregiver, a provider, clinician, researcher, and think about how we can be more creative in this as well. So thank you, everybody, and welcome to our event. This is in recognition of our planning committee, who is an excellent group of really hardworking folks who's been meeting almost every other week for several months to make sure that we can really fine tune the content, the invited speakers. And we're so honored to have several invited speakers who are joining us virtually via video presentation, as well as many of our live speakers as well. So thank you, everybody. It's been a wonderful team to work with.
Good morning, everyone. I'm going to echo my two co-presenters today and just welcome everyone, and just so excited to be here at our third annual summit. You know, we're just so happy that this has continued with the momentum, and we've always had so much support. So I'm going to get us kicked off today with some poll questions just to get a sense of who's in the virtual room. My name is Gina Curry, and I'm the director of the Office of Community Engagement and Cancer Health Equity at the University of Chicago Comprehensive Cancer Center. So now we want to know who you are, who is in the room, and who do you identify as, a caregiver or a clinician? Have you ever attended a conference in the past? Have you ever participated in a clinical trial? And so on. So we're going to go ahead and launch that poll. Can everyone see it? And it's several questions, so you will have to scroll down to complete all of the questions. We're starting to get some responses. Great, great, great. I thought it wasn't working at first. We're at 75% of the participants completing the poll. We're going to see if we can get some more.
All right, we're getting some good presentation.
We're at 86% of participants answering. See if we can get that last few. You will have to scroll down to see all of the questions and be able to submit. Almost there.
All right. All right, Gina, should we take a look and see what you think?
We still have some coming through, it looks
Oh, good,
like.
okay.
Yeah, we still have some coming through. That's why I didn't want to cut it off,
Perfect.
but we're at 90% and just waiting to see if this lasts. Yeah, it looks like it stopped now. So I'm going to end the poll and I'm going to share the results. So it looks like the survivors are representing today. That is excellent. And then you see patient advocates, caregivers, researchers, clinicians are next. And then have you ever attended a conference? So we have a lot of new first timers. That's excellent as well. We have 37% that said that they have not attended this conference before. And have you ever participated in a clinical trial? We have 31% no, but 12% have. No, but I would, 13%, that's awesome. What interested you in registering for the event? We have a tie with to learn about patient experiences and to learn about different types of gynecologic research.
Excellent.
You consider yourself a patient advocate? Yes, 35%, the majority considers themselves patient advocates. No, and not sure. So by the end of this conference, this summit, you should know if you're not sure. And finally, are you interested in being a patient advocate? We have 34% saying yes, and 16% saying maybe. So that is great. So I'll stop sharing.
Yes, I think someone mentioned many people do play many different roles in this space. And so I do agree that we will probably hear from some people who are playing both the roles of survivor and researcher or advocate and various different ways. So we're excited about having everyone here today for this. All right.
So here's just a look at our agenda today, and we're going to be checking off as we go. So if anybody pops in or off, they can see exactly where we are in the agenda. But Erica is going to be putting this in the chat box, so you can refer to it whenever you need to throughout the summit. But this is our very ambitious summit agenda, and it's going to be awesome. Next, we're going to be viewing a video montage from the Cancer Center leadership, Chicagoland Cancer Center leadership, but we are so proud to have a national representation that's going to be giving us a few remarks and objectives to today's summit. So we are just delighted to hear from the director of the FDA's Oncology Center of Excellence first, and then we'll hear from our area cancer center directors, our local cancer center directors, but excited to have this these national remarks.
Hi, I'm Dr. Richard.
Yep.
Pastor, and I am the director of the Oncology Center of Excellence at the United States Food and Drug Administration. And I'm pleased by the organizing committee to be offered the opportunity to speak to you at the 3rd Annual Chicago Gynecological Cancer Research Survivor Summit. This is a very important conference for several reasons. The primary one is that it brings together patients, researchers and other stakeholders to talk about research and ways for patients to be involved in clinical research and in the care of patients with gynecological cancers. And I think that this is an important thing to bring people together. I'm also very happy that this is occurring in the city of Chicago, although most of you are in a virtual situation here. But nevertheless, I have ties to Chicago. I'm a Chicago native. I did my medical training in Chicago and rotated through most of the major teaching hospitals there, including Northwestern, Rush Presbyterian St. Luke's, University of Chicago, Loyola, the late Michael Reese Hospital when it was in existence several decades ago. So I have a vast experience of Chicago medical hospitals and research centers there. And I think that this is a city that is so vibrant in the care of patients with oncological problems, as well as leading the forefront of research in oncology conditions. So you couldn't be in better hands there than Chicago. And I am so happy to have shared that experience in my early part of my career being in Chicago. In addition to that, I have personal experience with gynecological cancers, and that my late wife had ovarian cancer, and I served as a care provider and supporter of her during her years dealing with this disease. So I have personal interest in gynecological cancers and a personal experience with this, so I fully understand the journey that most people are going through with this disease. I'd just like to highlight certain issues here that I think could be coming to the forefront, and I've been given by the organizers some of the objectives of the course, and I want to go over those with you. The first one is to promote a bi-directional understanding of both the researcher and the clinical experience and the survivor experience. And I think it's very important that both groups really have a dialogue, learn from each other, because there's valuable information to be shared. The second objective is to promote an understanding of research topics, clinical trials, new areas of discovery between institutions. And I think that this is also a very important issue since, as I mentioned before, there are so many vibrant research organizations and institutions in the city of Chicago. It really this city really brings together a wealth of experience in gynecological cancers, as well as in the general treatment of oncological diseases. The third objective is really to promote an understanding of the role of the patient advocacy and research, clinical care, and legislative policy in gynecological cancers. And I think that this is very important. Here again, I think the most important thing that patients could understand is the need for education, and the education that they will obtain through this conference will also lead to really a stronger position in advocacy, as well as empowering them. And this is a central area that I would like to comment on, and that is empowering patients through really knowledge. We here at the FDA have several programs that are geared toward patients. We bring patients in to hear their voice, so to speak, at our oncology advisory committees where we make decisions on on applications. And in addition to that, we have various conversations on cancer. Part of the Oncology Center of Excellence, which I lead, which was set up about 5 years ago under the original Cancer Moonshot, really is to have a community outreach. And we have a specific project called Project Community, which is geared to really bring people into the FDA, not physically, but virtually, in our discussions about certain regulatory topics. So we would encourage you to stay tuned to the Oncology Center of Excellence website to see future programs that we may have that are really quite patient specific. In addition to this, the fourth objective of this program is really to support trainees in gynecological cancer research, to engage diverse audiences and further develop communication and scientific presentation skills. And I think that this is quite self-evident. One of the other major issues that I would like to address is the OCE, or the Oncology Center of Excellence, commitment to diversity in clinical trials. And we actually also have a separate program that is called Project Diversity, really trying to deal with and encourage sponsors to have a population of patients that are representative of the US population in their clinical trials. And this is a major goal of ours, which I hope you will also discuss during the presentations and the discussions surrounding this. So if I could just end this brief presentation by just mentioning a few words, and that is something that I've already repeated, and that is empowering patients. And I think that it is very important that we have a dialogue with the patients, because they are the ultimate consumers of what is approved by the FDA, but also have an interest in hearing from their diverse opinions regarding our approval decisions, cancer care in general, and where the future of oncology will go. So I really hope that you will have a very, very successful symposium here at this summit. Again, it's your third annual Chicago Summit, and I couldn't be more happier rather, I should say, than to have it in Chicago, my hometown. Thanks. Bye.
Excellent. Now we'll be hearing from some of our local leaders.
My name is Kunle Odunsi. I am the director of the University of Chicago Comprehensive Cancer Center, and I would like to welcome you to the third annual Chicago Gynecologic Cancer Researcher Survivor Summit. This promises to be a very exciting day because one of the main areas that we really want to begin to understand and focus on is for us to engage with our patients, especially in the area of research. Our Comprehensive Cancer Center has the mission of trying to understand, prevent, cure, and cure cancer. But we know that this requires tremendous research. In fact, over the past 30 years or so, we've made tremendous progress in gynecologic malignancies. The outcomes are a lot better. For example, the past few years, the PARP inhibitors and other forms of therapies that have come to help our cancer patients. However, there's still much work to be done. That work requires engaging with everyone, every stakeholder, including our patients and survivors. So we would like to engage with you to make sure that as we design research projects, we get your input, your insights. We also have with us today some students who are conducting research, and this may well be their first encounter with patients. So this is also a very useful training opportunity for some of our trainees. Again, I just want to welcome you. I think today promises to be a great meeting where we will learn from you, where we will engage with you, and hopefully some of you might even consider becoming advocates for research, for gynecologic cancer research. So thank you and have a great time.
Hello, I'm Leon Platanias, director of the Robert H. Lurie Comprehensive Cancer Center of Northwestern University. It's an honor to welcome you to the third annual Gynecologic Cancer Summit, focused on increasing understanding among cancer survivors and researchers. At Lurie Cancer Center and across Northwestern medicine, Our gynecologic cancer team is committed to narrowing the gap between the laboratory and the clinic. We all benefit from creating opportunities like this one, enabling cancer survivors, scientists, doctors, and healthcare professionals to build strong, collaborative partnerships. Sharing knowledge not only fuels research and clinical trials of promising new treatment options for women with gynecologic cancers, but it also empowers us to improve their quality of life. To me, the importance of research is how it affects the patient. That's how we can have the most impact now and in the years to come. Thank you. Hello.
Hello, I'm Dr. Jan Kidieski, Director of the University of Illinois Cancer Center. Welcome to the third annual Gynecological Cancer Researcher Survivor Summit. Today we are honored to be among our partners, the University of Chicago and Northwestern University, to support the summit's objectives of multi-directional exchange between patients, clinicians, and researchers. We function as a community-to-bench cancer center that creates space for patients to be at the center of decision-making in their care, in clinical research, in clinical trials, and in patient advocacy. We aspire to be at their side every step of their journey. Once again, welcome and enjoy the day's presentations.
And so I am going to not spend very much more time on the subject, summit's objectives, because Dr. Pastor did such an eloquent job. But I do want to say this briefly. We want to make sure, because we have so many survivors on today's summit, that it's incredibly important for you all to engage with the oncologists, as well as with the junior researchers and with one another, as we do most of our work together through our breakout rooms. And so having said that, I'm going to remind you that this is bi-directional. We want as much feedback from you as we possibly can. Number one, for you to have the best experience, but number two, for us to know what to do better for the fourth annual, summit. So with no further ado, I'll send it over to my partner, Dr. Lee, to talk about the critical areas in gynecological cancer research.
Thank you, everybody. I am so pleased and honored to be able to have invited and have many of our key leaders in GYN cancer. These physicians that you see here, Dr. Tanner, Dr. Yamada, Dr. McLaughlin, Dr. Leota, are our section chiefs or division directors of our gynecological oncology sections throughout the Chicagoland area. And many of you guys may know them as mentors or your own patients or your own physicians. We want to talk a little bit and get their input and their perspective. So the questions that we want to ask, and I'm hoping I can see everybody at the same time, is that I'm going to start by asking everyone a couple of these same questions so that we can really kind of get a discussion going about some key topics around research. So maybe I'll start in the row that we're here. I'll start with Dr. Tanner, and go through everybody as I call on you. I know it's a little bit hard on our combined Zoom, but to kind of think about, as we're embarking on this event, what are levels and ways that you and your team as a whole engage patients and survivors in both clinical care, improvements, or in research efforts? We'd just like to get a sense of how you view that and what everybody is doing.
Are we going in order?
Yep, I'm going to pick on you first, Dr. Tanner.
Sure. So, for the first question, engaging patients and survivors in clinical care, I think is in some ways straightforward. I mean, obviously we always want to listen to patients in clinic and and get their specific feedback about how we can do better. There's, very straightforward things like, we read through every comment that patients provide about their experience in clinic and address when there's areas to improve. Seriously, I look through every comment every week to make sure that we're taking great care of patients and that we're addressing issues as they come up. More broadly, when we engage in events like this, or we have our Survive and Thrive conference on October 7th, and you can, on my Twitter feed, you can find some details about that. But when we have an event like that, we engage with patients about what kinds of topics they want to hear about, and I think that extends both to like research topics that they want to hear more about, and ways to improve care and survivorship. So those are more structured ways to address it. And I think that from a research standpoint, in some ways it's hard because we clearly have the responsibility of trying to figure out the trends of promising research because we have the training for that. But certainly many times I've had situations where patients have brought up to me trials that they've found, especially early phase trials at other places. Sometimes there's research going on. It's so hard even for us to keep track of everything that's going on. And then, we can figure out whether that's something that we can add to our pipeline of studies. And, there's a long process of getting studies open. So we know that patients want access to trials, very consistently tell us that. And so it's really our responsibility to make sure that we're opening trials and keeping options available for patients, and that we don't have any gaps in trial availability over time. So that's a complex process, but we always welcome patient input from that.
That's fantastic. Thank you. Dr. Yamada, what are your thoughts about how or different ways that you think of in terms of engaging patients and survivors in kind of this space that we occupy between clinical care as well as our research efforts?
Thanks, Neetha, and thanks again for putting together this conference. It's really fantastic to see everybody. So, yeoman's work. So I think that the way that I look at this is that the trials that we do and the clinical research that we roll out really has to be applicable to the patients and meaningful to the patients. And it has to be accessible, and I think it has to be understandable. And I think we need to kind of reevaluate, what the endpoints are of the trial. So I think, there has been talk about, and we've certainly tried to do this at the University of Chicago to try and bring clinical trials out into the community where people are. The trials are so incredibly important, but they can be very labor intensive. And so are there ways that we can make them more accessible to patients by either bringing them closer to where they are or be a little more understanding and make things easier? Can we incorporate telemedicine visits, for instance, or easier ways to do lab draws or radiology studies to make it more accessible? I think the other area is that we have to make them understandable to patients. So I recall probably 10 years ago, some of the consent forms for the clinical trials were like 35 pages long. I mean, I can barely go through a consent form that's that long and be able to feel like I'm really explaining that to a patient. So I think that we need to make sure that we're capturing what's meaningful in the trials to be able to present it to patients. And I think that, we have to establish trust with patients as well. And so I think that having clinical research teams that really take stock of what research is going to be meaningful to the patient population that we're taking care of, and also that we have clinical research staff that are representative of the community that we're taking care of, so that there's a level of trust that's built there, I think is very important. And then I think we need to reevaluate the studies that we're doing and look at the outcomes and look at things like toxicity. Are the toxicities manageable? Are the response rates and the toxicities that are there, are they going to balance out? Do we have the resources to be able to help people to do that? And then the last area that I think has been interesting has been, and one of our fellows is kind of looking at this, is the financial and time toxicity of the trials. It's something that's, that we need to be able to kind of explain to patients because that's going to be important for them. So the patient-centered research, aspect is very important. I hope that answered your question.
Yeah, I think so. Thank you. Dr. McLaughlin, if you want to go ahead and continue with your thoughts about engaging patients and survivors in different elements of care and research.
Thank you, Dr. Lee. I just first need to take a moment to to say how honored I am to be on this panel of my esteemed colleagues in the city who are also, for the record, leaders in the field of gynecologic oncology. So I'm really excited to be here today. And I echo everything that you heard from Drs. Yamada and Tanner about representation and patient-centeredness and having the right studies available with the right treatments at the right time for the patients who you serve. I would add that at UIC, we have done a few things in direct response to this conference. And I was an audience member in this conference in year one, and it changed everything we do at UIC in the gyn cancer world, starting with including patient advocates and survivors early in the pipeline of trial development. Any study that I do or that gets built in the gyn cancer space that's investigator initiated, that study goes by the eyes and the ears of a patient advocate and or survivor or a team of them to make sure that we're not missing the mark. We've also included Kimberly Richardson as a representative on our disease team, which is the board within the regulatory process at UIC that looks at every clinical trial that we're going to open up. And we are able to discuss it from the patient standpoint as well. It's been really valuable to have that representation in the pipeline of development. And from the clinical standpoint, and this is a research initiative as well, and it really, I'll just circle back to Dr. Kirievsky's comments about our community to bench approach at our cancer center. We have a project opening up recently funded by our cancer center's Office of Community Engagement that is going to help us develop an interdisciplinary integrated approach to cervical cancer treatment. The big model is to engage community stakeholders as part of the development of this clinical enterprise. The recently funded project, excuse me, is allowing us to recruit community members from the 5 community areas in very close proximity to our cancer center who have incredibly high mortality rates from cervical cancer, and bring them to the table and talk to them about their community experience and their community expertise. We recognize that our neighbors are coming from backgrounds that we may not understand, and we may not just be able to say, okay, our doors are open, come on in. And so I'm really excited that our cancer center is supporting our initiative to really roll up our sleeves and get in there and see how we can learn more about how to provide better care for a cancer that shouldn't exist anymore.
Thank you, Dr. McLaughlin. It's fantastic perspective on engaging community and thinking about those really shared goals and how do we establish those shared goals. So Dr. Leota, thank you for joining this morning. I'd love for you to kind of go through what your thoughts are about patients and survivors in your care and research efforts as well.
Good morning, Dr. Lee. Thank you so much for having me on this panel. I've also been participating through this project since the beginning 3 years ago, and it's just been a pleasure to see how much it's expanded. And I know my patients have benefited from participating, so I really appreciate everyone's efforts for creating this wonderful yearly meeting. It's hard to go last after everyone said all these wonderful things. I'm going to echo everything that you have all shared. Dr. Tanner, I know you commented on the open communication with patients, and I agree that, you know, I've had conversations with patients and they've opened my eyes to new studies and things like that that I didn't realize were there. There's just so much out there for us to try to organize for patients. So, you know, to have a patient that's comfortable talking with you and having that open line of communication, I think is important, which is is a reflection of all the healthcare team members, the nurses, the advocates, et cetera, so that the patients feel that they can get their questions answered and perhaps develop or help in developing their own treatment plans. As far as survivorship and engaging patients, Loyola actually had a unique opportunity a few years ago to work with a patient, ovarian cancer survivor, who was also a filmmaker. And we collaborated with her in a short film that she made called 1 Year Later. I'm not sure if anybody's seen that or not, but it's a pretty powerful demonstration of a cancer survivor who decides to go to Italy for after she's finished her treatment, because in the family realm where she's been thought of as a patient battling cancer. And so she goes to a new place where she becomes a person who's had cancer. So moving into that new phase of life, which I think is hard for a lot of our patients to cross over to. And we've been using that to help with cancer survivors and also in research efforts to sort of generate funding for more cancer research.
Thank you so much. And then I'll make you go first on the, we'll go on the way backwards, since we're going to be, we don't have as much time, since I've loved hearing about everything. What do you think of in terms of like, what are challenges in gynecological cancer research, and also the areas that you're sort of most excited and hopeful for? So I'll kind of combine our questions 2 and 3 to make up some time, but what are your thoughts about that?
So definitely challenges in research is I think we've discussed some of this as well, but sort of making patients aware and trusting of doing research, because I think sometimes when we talk to them about research projects, they might feel that they're like guinea pigs or being tested on, et cetera. And I feel that stigma needs to be dispersed so that we can get patients enrolled and feel comfortable enrolling in studies. Also, the need to increase diversity in the participants in our studies, paying special attention maybe to patients who don't speak English, making sure that we have consent forms in different languages and able to engage patients in that regard so that we get a more diverse population. And then I guess I'll move on to some areas that I'm excited about. I think gynecologic cancer development right now is very exciting, especially in the area of personalized medicine and developing treatment plans with molecular profiles instead of just other studies. Like, for example, the molecular profiling that's been identified in endometrial cancer can hopefully help us as physicians develop treatment plans for patients on the molecular level, and also incorporating the clinical pathological risk factors. So we hopefully would have a more personalized treatment plan for these patients.
That's fantastic. I totally agree with you. Dr. McLaughlin, I'm going to go across my screen this way in terms of things. What are your thoughts about challenges as well as areas that you're most excited about?
Well, first, 100% everything that Dr. Liotta said. I would add to that I feel like all of the challenges in research for gyn cancer are rooted in awareness and advocacy or lack thereof. Gynecologic cancers are not maybe as common as other cancers that have a lot of awareness and a lot of advocacy. And ultimately, those things drive funding and those things drive awareness. But when you have patients in your clinic who say, what do you mean you're seeing patients with cervical cancer all day? We don't even have our own ribbon. That is an area of opportunity for us to do better so that downstream these other challenges are less challenging. I like to lean into the hope question a little bit more because I'll start by saying that I stay in academic medicine specifically because it brings me hope every day. You get the hope of the fulfillment of working with patients and recruiting patients from your clinic to become advocates, and you get the opportunity to see students become physicians, become researchers, become leaders in gynecologic oncology. And I will just say, as a proud mama bear moment, that I have one of each of those on this call today. And I just am so excited by it. I think that gives me hope. And events like this coming together, just seeing the change in the last 3 years and the impact and the power of having an FDA leader give such a remarkable opening for us, that gives me hope.
Thank you. Dr. Yamada, as we scroll downwards here across my list, what are your thoughts about challenges as well as the things that you're excited about?
Well, I think Dr. McLaughlin and Dr. Leota have done a great job kind of framing what is really important. I think one thing that is, just to pick up on the awareness piece and the advocacy piece, I think, one of the things that we did as part of the Society for Gynecologic Oncology last year was to really raise awareness about the funding, the national level funding that goes toward gynecologic cancers. And, concernedly, like endometrial cancer mortality, for instance, is increasing. It's one of the cancers where mortality is increasing, and yet the funding at the NCI level for that cancer is abysmally low compared to some of the other cancers. So I think that that's one area where we can really advocate, and we have advocated at the national level and the congressional level to improve, like Department of Defense funding, to improve funding for people to be able to do the really meaningful research and have the time to do the really meaningful research that will kind of push these areas forward. So I think that that is incredibly important. And then I just think, we're in such a unique position as gynecologic oncologists. I think everybody on this panel does surgery, gives chemotherapy, is involved in clinical trials, dovetails with translational researchers to try and identify those areas. And so we do a lot of things. And so supporting, I think, our young investigators to be able to have the time to interface and give them platforms to interface with people, researchers, survivors that are really going to identify the really meaningful areas of research, I think is important and something that I'm really hopeful for. But I really want us to improve the awareness of the level of funding for gynecologic cancers. It has to be first and foremost, especially with the mortality rates that we're seeing.
I think you're totally right. And I just applaud. I think Shannon had a shout out to you here, Dr. McLaughlin, about your leadership through the Society for GYN Oncology as the president this past year. This is the first time the Department of Defense actually funded endometrial cancer, which is huge in terms of not having that stream of funding and the legislative and the work that you had to meet with a lot of leaders and everything we really appreciate. So Dr. Tanner, what are your thoughts as we think through sort of challenges, but also, again, exciting areas for us to be thinking about?
Yeah, so I, again, echo what everyone else has already said. And I think the things that have already been said are probably the most important ones, but just to create just a little bit more of a point about it, the outcome of the research funding from 10 years ago is what we're seeing in the phase 3 trials now. It takes many years for the effort and the support to be realized. So that's why it is so important that we continue to press for more funding, because a lot of those efforts, it takes years and years for things to reach even phase 1 trials, let alone to phase 3 and approval of new drugs. So that's really important. And I would also add that sometimes there are things that we don't really necessarily expect that become barriers. During the COVID pandemic, we've seen a huge issue with labor issues with, like, in terms of having enough staff in cancer centers. And I don't think that's something that necessarily many of us really thought about as being a major barrier, but we have to have coordinators and regulatory staff to make sure that studies are, that budgets are put through, and that there's a huge amount of infrastructure that's required for that. And sometimes those things can be really, really serious issues, and they require working on not just like the research side, but on like administrative side. We need really effective leadership on that side to make sure that we can effectively put trials together. And I think for many of us, that was something that was a surprise in the post-COVID era, is that we have to have the support staff to do all those things. So hopefully a lot of those things will get better in the coming years, but you never know what the new challenges will be, but we're always trying to figure them out and move forward.
Thank you so much. I think you're right. I think it's about building that whole team, like you're saying, and really us realizing how much of that infrastructure needs to be built, supported, funded, and also, as many of the other speakers have mentioned, representative of our patients' perspectives, as well as the diversity of patients who are diagnosed with GYN cancer. And so I'm thrilled to have all of your perspectives here. And I'm going to turn it over to Kimberly Richardson, so that we are going to hear now from our lived experiences of our cancer survivors, so that they can also share their expertise in what we are going to be talking about today.
Well, thank you. Thank you, Dr. Lee. As everyone here on the call knows or should know, I am also in 2013, was diagnosed with a rare form of ovarian cancer, granulosa cell tumor. And as a patient advocate, I try to make sure that we get an opportunity through this event to hear from survivors of all of our gynecological cancers. And so we're going to start off with Jen. We have 3, and each one of them will introduce themselves, and then we'll probably, if we have time at the end, have a question where we can do a roundtable. Jen, if you are ready, take it away.
Good morning. My name is Jen Swikowski, and I am a 3.5-year survivor of ovarian cancer. Back in 2019, after about 8 weeks of having what are now considered the typical signs for ovarian cancer, I had many weeks of bloating, abdominal pain, and frequent urination, to the point to I was going about every half an hour. After my primary care doctor had ordered a CT scan for me, it was discovered that I had ovarian cancer. So I had found my doctor, my gynecological oncologist, Dr. Langell. At that time, my treatment plan was to go for 3 rounds of chemo, have a debulking surgery, and then after that, it was 3 more rounds of chemo. And at that time, that worked wonderfully. I was NED for 11 months. Unfortunately, when I went back that next year for my 1 year, almost 1 year, I was having a recurrent cancer. So at that time, I went through 7 rounds of chemo again. We took a break for 4 months. And after that, cancer came back again. So actually, that's where I am right now. I was going through chemo at the time for every 4 weeks to every 6 weeks. And when I was diagnosed as a stage 3C. So with that, that's a later stage that comes with more of its challenges. But I just found out the other day now, as I have been going through chemo, that I just had 2 new nodules that appeared. So right now I'm in the midst of stopping chemo, and we're going through a new plan of action. So I will find that out in the next couple of weeks as to which way we are going. Sometimes it gets to the point to where what you're doing is no longer working. So we're on our adventure again to try to see what is going on. But I've actually felt very good through all of this process. I've done really well with all the chemos. No adverse reactions to anything. I can't say it's been a walk in the park, but it's one of those things where you keep your head up, keep a very positive outlook on everything. And I have been getting very involved with NOCC and just being a patient advocate for all those out there with ovarian cancer.
Thank you so much, Jen. I think that it's really important for people to know that while we're going through treatment, we also have internal hope. And I love the fact that you called it an adventure.
Yes.
So thank you for sharing your lived experience with us. Shakia, if you're on the line, you're next.
Hello, everyone. My name is Shakia Allen. I am a uterine cancer survivor. I was diagnosed in 2009 with adenocarcinoma in the uterus or in the endometrium. And I had several symptoms leading up to this, which they went ignored. And so finally, after years and years of going back and forth to gynecologists trying to find out what was going on with the abnormal bleeding back in 2009, I was finally diagnosed with the endometrial cancer. Due to lack of information, the treatment method that I chose was a hysterectomy, and that was the treatment option that seemed easiest, or I guess that the oncologist had the most information about. They did leave my ovaries, and sadly, last year, I started to get cysts on the ovaries, and so now we are going through 3 months of, evaluations every 3 months, trying to make sure that that does not result into ovarian cancer. So I just wish they would have taken everything. I know they were trying to help with the menopause symptoms, but I wish they would have taken everything, because now I'm having to deal with this. I'm very thankful to be a survivor. So what I did was I started my own 501c3 to be a patient advocate and make sure that the awareness is being put out there for endometrial cancer for all races. And I do appreciate you guys giving me the opportunity to talk today. Thank you.
Thank you, Shakia. Well, we've heard one testimonial based on hope. Shakia's was based on gratitude. And so we're going to end with Tristan. Tristan, come on the line.
Okay. Hello from the middle of Canada. I'm Tristan, and I am a clinical oncology social worker actually up here in Canada, but I'm also a transgender man and a stage 3C ovarian cancer survivor. And I know I'm a bit of a unicorn, so that's why I am sharing my story as many places as I can. It's been about 19 years actually. I had my cancer surgery at 30 years of age. I had symptoms all the way from my teens. My menstrual cycle was not regular at all. In my early 20s, I was diagnosed with polycystic ovarian syndrome. Both ovaries were covered to the hilt, but that entire time up until my diagnosis, the physicians kept writing all my symptoms off to irritable bowel syndrome. And it wasn't until I demanded an ultrasound and further investigation from a doctor that ovarian tumor was discovered. And when I had my surgery, when they opened me up, they realized that it was a low-grade proliferating borderline serous tumor, but it had obviously been going a long time, because they said these are the tumors. The extent of my tumor was the same they've seen in people that are in their 70s or 80s. And so I had a full hysterectomy, double oophorectomy. The tumor was climbing toward my bladder, my liver. The main right orbital nerve got completely severed. So they had to bring in a neurosurgeon to stitch me up, and I had to kind of learn how to walk again. Took a couple of years, but at the time I was presenting as a woman. A few years ago, I started my transition, and that's why I'm the dude sitting here today with the beard. And back then, after I had my surgery, I was referred to the local cancer center, and I met with a medical oncologist, and I didn't know anything about cancer. And I remember saying to him, "Now what?" And he said, "Well, you go live your life and you tell us if you have any abnormal symptoms." And I said, "What are abnormal symptoms?" And he told me what they were. And I'm like, "Those are all symptoms of IBS." and I have it. So I was kind of lost. I will also quickly share, I did attend some of the local ovarian cancer support groups, but I've been trans all my life since I was little. I'm one of those trans people that I knew when I was really little. So, sitting in the room with the other ovarian cancer survivors, I was coming from a different place. I was relieved, relieved of the parts that were causing me gender dysphoria for all those years. So it was a really different experience for me. And now as far as I know, I'm still cancer free, but I have had CA-125 tests the last couple of years, and there's some indicators there, but I'm mostly here in this capacity because now I'm in the world of being an ovarian cancer survivor as a transgender man, and what that looks like, accessing those spaces that are, cisgender women dominant, of course, understandably, and I'm really happy to be here. Thank you.
Thank you. So Tristan, that's bravery. So it seems to me that throughout every one of our experiences, we have to have all of those characteristics. We have to be hopeful in the midst of that diagnosis. We have to be hopeful. We have to be grateful for the support that we're getting from our cancer care team, as well as our families. And then we have to be brave sometimes, right? So I wanted to make sure we got an opportunity to ask you guys one question. And it's one question, and I'll go just down the line. And Shakia, the question that I have is, what would you like to tell oncologists about patient-provider communication?
What I would like to say is I believe that patient interaction with your gynecologist and your medical team is most important. You have to have that open dialogue with your doctor, and then your doctor has to take the time to actually listen and give you the opportunity to answer or ask as many questions as possible. I bring a journal and I have tons of questions, and sometimes the doctor doesn't have time to answer them all, but I believe that if we take a little bit more time to actually interact with our patients or with the patient will be, I guess, more apt to speak to you. They won't feel uncomfortable bringing things up. And so I believe that having that open dialogue with your doctor is very, very important. You have to feel comfortable with your doctor, and it's the doctor's responsibility to make sure that their patients are comfortable asking anything.
Thanks, Shakia.
You're welcome.
And now, Jennifer.
Yes, I have to say, in terms of that, with my own doctor, I am very comfortable. University of Chicago does have a program that is that we are all made to be aware of, is to speak up, and they always remind us, and they always ask, are you aware of the Speak Up program, so that we know that we can speak to our doctors, nurses, chemo nurses about anything that we need. And I know it's key to ask whatever we want. Also, I know it's very good that my doctor allows us to email him as well. I have a phenomenal chemo nurse that communicates with him as well if I'm not able to get him right away. But knowing that you can count on your doctor or his support team in order to have yourself heard. And I definitely do feel heard, but that is definitely key, because if you don't feel comfortable with your doctor or his staff, then you need to go elsewhere.
Indeed. Tristan.
Two things. I'll share from the clinical oncology social work side, but also personal experience side. It's time, like Jennifer said, time. When you have that experience and you're in your new patient consult, that takes a long time usually. And then a clinical trials nurse, I don't know how it goes down there, comes in, but it can be like a 4-hour appointment. I really hope that we can find a way to have more than one appointment at least, so that everybody doesn't get all at once. The other thing I'll share too is not to make assumptions about what these body parts mean to people. And that's where I come from, is a lot of assumptions were made, and I was much younger and less assertive than I am now, but how important it is to find out who that person is right in front of you, that patient right in front of you. Who are they? What's meaningful to them? What are their goals? And that takes time to have a good, thorough conversation about. So.
Well, thank you. Thank you all. I know from my own experience, I've learned from working with oncologists like Dr. Lee and Dr. McLaughlin, seeing them as human beings as opposed to just doctors, and getting that opportunity for me to tell them about my own personal experience. It became more of a therapeutic journey for me, so that I began to see things from both sides, right? Because sometimes as patients, we are like, well, that oncologist said this to me or didn't say something to me. And we often don't get that opportunity to step back and see that they have several patients they're going to see and interact with so many different people, and they're human. And so sometimes we do need to get that opportunity to get that perspective, to see things from that other side sometimes. So I thank you all for sharing your lived experiences with us today. And I am going to transition us to our next session, and that is our breakout session. And Erica, you're going to explain to everybody what we need to do next. Thank you all, survivors.
Hello. Yes, good morning. My name is Erica Rodriguez, and I'm the Latinx Outreach Specialist at the Office of Community Engagement at UChicago Medicine. So we are going to move on to our first breakout session, and in which we will be discussing possible collaborations between survivors, advocates, and investigators. Everyone will be placed in a room, and we will share with a moderator. We will be sharing those salient points of each room at a later time this morning. So if I believe you guys will all go to your rooms.
Such a great group. I hope everybody's discussions and connections were as great as ours. I'm really excited. At some point, I do want to take some giant Zoom screenshot, but I'm not really sure how to do that. So I'm Googling on the side how to take a screenshot from my Mac. So, all right. I think we are moving on to hear from Dr. Kernett about trials and types of gynecological research. So we'll have Dr. Kernett, I believe, share screen and get going.
All right, thank you. I'm gonna start sharing my screen. Can everyone see okay? Yeah.
Yes.
Yeah, perfect. So I'm Katie Kernett. I'm one of the gynecologists at the University of Chicago. I'm really excited to be here. Thank you for letting me participate again. This has been a great experience the last few years. So I'm going to spend a few minutes just talking about gynecologic cancer research, clinical trials, and I'm going to specifically focus on clinical trials and some of the phases and what that means, because I think that is a really important kind of terminology and information to understand for patients and families and anyone interested in research. So, why do we do clinical trials? And a big part of this is because we all want to know more. We want to increase our knowledge, but clinical trials aren't a silo. They exist in this kind of framework of a lot of other types of research studies, case reports, cohort studies, case control studies, basic science, translational, lots of other designs that kind of build the foundation long before you get to a clinical trial. So we're always looking to expand our knowledge, and clinical trials in some ways represent kind of the final frontier. So we want to find new treatments, and there are all sorts of other types of clinical trials, interventions, other outcomes we can look at, but we really want to find new treatments for cancer. That's a really big part of it. We want to do it in a way that's safe and controlled, and that everyone is kind of taken care of so that we're not doing anything reckless or foolish. And that's where the basis of clinical trials really come in. So going through the phases of clinical trials, phase 0, and these headings that I got came right off of cancer.org. They have a wonderful patient-facing kind of information there, and I have some resources at the end that I send patients and families to in my own clinic. So that's where some of this came from. But phase 0, this is exploring how a new drug may work. An example would be, taking a very few number of patients, giving them a very short course of a specific medication and then doing a biopsy or doing a surgery, something that they would have gotten anyway, and looking and seeing what that medication or treatment did to the tumor. So understanding the biology in a more kind of real world, but very limited setting. Phase 1, the goal here is to see really, is the treatment safe? This is a small number of patients. I usually think of somewhere around 20 to 30 for most traditional phase 1 clinical trials. The goal here being to understand the side effects or the safety, the toxicity of these new drugs, but also the dosing of the drugs. So an example would be that, you know, somewhere around 30 patients will be enrolled. They're in usually small groups and they're given different doses. And then we do a whole bunch of statistical designs to figure out how many patients have to have how many toxicities of varying degrees to decide what's a safe dose. And then at the end of all of that, you walk away and you say, okay, for this drug, whether it's acetaminophen from many, many years ago or a brand new cancer drug, what is the dose that we should be recommending that people use? So phase 2 is getting into the question of, does treatment work? And this is usually a little bit bigger population. I would say maybe 50 to 100, but it can be a little bit bigger or a little bit smaller. This is usually in a more focused population. So maybe patients with ovarian cancer or patients with cervical cancer. Rather than in phase 1, a lot of times they'll be a little bit more broad. Patients with any cancer, patients with gynecologic cancer. And so this is, we're starting to narrow the field a little bit. But we're using that dose that we decided from phase 1 and moving it into a group of patients to see, does this dose have the effect that we want? Does it seem to shrink tumors? Does it, is it really as tolerable as we thought it was going to be based on those smaller studies we already did? So this is a more focused kind of question of, does the drug work? And then phase 3, this is when people think about clinical trials. This is really what, this is what a lot of people will kind of have in their mind already. This is the most, I guess, famous type, but also one of the rarest types because they are so big and so difficult to actually get up and running. The question here, and this is the final step before a drug becomes available, so to speak, is whether this new treatment is better than what we're already doing. So if we already have this chemotherapy, like we already have carboplatin and paclitaxel, if we're going to use a new treatment, we have to prove not only that it works, but that it works better than what we're already doing. So an example of this would be, you know, a larger number of patients, and hundreds, sometimes thousands. These are very big clinical trials that we design very carefully. We have to know what our standard treatment is, and then we have to decide what are we going to compare and how are we going to compare it. So for example, if we have 1,000 patients who with endometrial cancer, half of them are going to get the new treatment, half of them are going to get the standard treatment, and they're going to be randomly assigned to those groups. So we don't get to choose. And this is a really exciting study design because we get to, you know, define the new treatments. But this is also one of the downsides here for patients. You know, I can see the disappointment sometimes when they enroll in a phase 3 study and then they get assigned to a treatment that they would have gotten if they weren't on the clinical study. And so these are some of the important caveats, the pros and the cons of some of these really important studies that we do. And then phase 4, you know, after phase 3, hopefully the drug works and it is better. The drug company usually will go to the FDA and ask for it to be available to all patients and insurance companies. Phase 4 is kind of after that. After that drug is approved, what else do we need to know about it? And this is basically thousands, tens of thousands, many, many, many people. And it's really the real world setting of how does this drug work? How does it work in patients in real places and doing it in real time? So let's say there's a brand new cancer drug and any patient who's prescribed it goes into a registry in a database, and then they follow them over time to see what are the side effects, what happens if you're not doing these lab tests every single week like they did in the study, kind of what happens when we're doing it the real way. So these are some resources that I've shared with patients before that I think is really are really helpful and accessible. And what I want to do now, sorry about that, is introduce our next speaker kind of with this all in mind. I find that a lot of patients and family members voice some unease about joining research studies for very understandable reasons. I think a big part is that we don't always talk about all the safeguards and planning that exist that go into these study designs. So our next speaker is Dr. Lindsay McNair. She's the Chief Medical Officer and IRB Specialist for the WIRB Cooperative group. And she will explain a little bit more about the protections that exist for all clinical trials.
Thank you, Dr. Carnett. I'll put my slides up. And can everybody see them?
Yes.
Okay, thank you. So thank you very much to the organizers for inviting me to be here today. It's a pleasure to speak to such a group with such a great purpose and for such a good reason this morning. I'm going to talk a little bit about how research participants are protected and the systems that are in place to do that when people are participating in clinical trials. So I'll start with one of the fundamental documents that provides us with a code of research ethics that informs how we make decisions about how clinical trials are designed and conducted. There are a couple of other ones, but we'll focus today on the Belmont Report, since this is the one that provides the basis for the regulations that guide the way we do research in the United States. So the Belmont Report was put together by a specific commission, which was founded in response to the publication of the Tuskegee syphilis studies. And I don't have time to go into the history of the Tuskegee study today, but when that became public, there was a bioethics commission that was convened, and they were asked to create a code of research ethics that could be used to guide the conduct of research in the United States, although these principles are adopted by many other countries as well. And what they published after 4 years of working on this question was the Belmont Report, which is the ethical principles and guidelines for the protection of human subjects of research. And the basis of that report, it's a great report to read. It's only about 7 or 8 pages long. It provides some really interesting discussion of these concepts. And there are 3 basic principles that should govern the conduct of research that involves human participants, although they use the term subjects in those days and in the regulations, which is why we still refer to subjects sometimes today. So respect for persons, which is the concept of autonomy, and that each person should be able to make decisions for themselves. The concept of beneficence, which goes even above do no harm, which is non-maleficence. But beneficence means that we should be actively seeking to try to do good in the research that we do, not just not to harm, but to promote good. And the concept of justice, which is really distributive justice, which means that the risks and benefits of research should be evenly divided across the populations, and no specific group of people should be used because they are convenient to ask them to participate in research, unless they will also receive the benefits of that research. And then in the 1980s, those principles that were described in the Belmont Report were then kind of adapted into the basis of our regulatory system for oversight of human subject research. So for federally funded research, that's governed by 45 CFR Port 46 in the Code of Federal Regulations. This is the policy for the protection of human research subjects, subpart A, which is the main basis of regulations. This has subsequently been adopted by multiple federal agencies, the Department of Defense, the military agencies, not the Environmental Protection Agency, they have their own code, but about 19 different agencies, I think, have signed on to these regulations now. So it's referred to as the common rule, because it's common to all of those federal agencies that fund the conduct of human participant research. There are some subcodes that have additional protections for pregnant women, children, and prisoners. And then for FDA regulated research, very similar, but a separate set of regulations that governs how any research that is used to support the development of new drugs or new medical devices. And those are listed here as well. So that's for FDA regulated clinical research. Now, what those regulations cover is that both OHRP and FDA regulations say that an IRB, an institutional review board, must approve the research plan before the project can begin, before anybody can be enrolled in that research. This IRB, and I'll describe who sits on the IRB in a second, the IRB approval is only good for 365 days. That's the number that's in the regulations, which make leap years a little bit tricky, but 365 days. And must be re-reviewed at least annually to make sure that the research is on track and the researchers are doing what they said they would do. Sometimes if the research is more risky, the IRB requires approval more often. So I've seen studies that said every 3 months or every 6 months, you have to come back to us and tell us how it's doing. And then while that research is going on, the researchers also have to report any unanticipated problems that happened during the research. So anything that was not anticipated when the research was started that represents unexpected risks to participants, and that can be risks that are related to safety and adverse events. It can be things like breaches of confidentiality if records get lost. It can be other types of risk besides strictly medical risks. And also any non-compliance with the protocol or plan has to be reported to the IRB. So if someone was enrolled in this study, but they hadn't given informed consent yet, if there was a study visit that was conducted out of window, if there was a blood test that was missed, if those represent risk to the participants, those have to be reported to the IRB as well. IRBs have to have a minimum of 5 members, and they are designed so that they represent the institution where the research is being conducted, as well as the community that is going to participate in that research. So at least one of those 5 members must be a scientist, and if it's FDA regulated research, they must be a physician. At least one of those 5 members cannot be a scientist, so it can't have 5 scientist members. At least one has to be a non-scientist, and one has to be unaffiliated with that institution. So someone has to be from outside the institution. They don't work there. They don't have a spouse that works there. They aren't tied to that institution anyway, so that they are representing the community outside that organization and the views of the community. Now, most IRBs have more than 5 members. As an independent IRB, our base core membership is 9 at each meeting. I've been on academic IRBs that have 20 or 25 members to have people with lots of different specialties. But it becomes unwieldy at that point to try and get quorum for your meetings when you have that many people sitting on your board. And the boards have to include members who have the necessary expertise, scientific expertise and therapeutic expertise, for the types of research that they review. So if a hospital is a center of excellence in a certain type of cancer, certainly people with expertise in that certain type of cancer should be part of the IRB to help review those research proposals. But the IRB can also pull in consultants whenever needed to bring in that additional expertise if they need to. The IRB decisions are based on majority vote of that committee. Now, there are different levels of risk of research and different levels of risk of how the IRB reviews that research. And Dr. Kernet touched on this a little bit in her initial slide, where she showed you some of the different types of clinical studies that go on in addition to clinical trials. So there are some studies that are such low risk that they are considered exempt from IRB oversight. And those would include things like research that's being done using data sets that have already been de-identified. They can't be linked back to any specific person. There's no way to link back to who that information came from and no confidentiality risk. Or even studies that may not be biomedical studies, like observations of behavior in public settings. So if someone wanted to sit in a train station and observe what people did while they were waiting for trains, that kind of research can be exempt because it's very low risk to the people who are involved in it. Then there's research that is considered minimal risk, and minimal risk is no more risk than the activities that you would encounter in general daily life. So this includes some of the studies that Dr. Kernet mentioned, like case control studies or cohort studies, which are examples of observational study designs. There's no intervention being given, there's no drug being given. It's either doing interviews with people or getting data from medical records. But there's no intervention or study drug or anything like that happening in those studies. This can also include studies where we're looking at a new cancer biomarker, and the request for participants is, can you give us an extra tube of blood when you come in to have blood drawn for clinical purposes anyway? So the risk is really just the risk of that additional tube of blood. So that's pretty minimal. And those can go through a level of review at the IRB that's considered expedited, where just one experienced IRB member can review those proposals. They don't need to go to the full committee. If a study is considered more than minimal risk, they do go through the review by that full convened committee and discussion of that full committee before they can be approved. And more than minimal risk studies really involve any clinical trial that has an intervention. So new drugs, even if the safety profile is really good, just the fact that there's an intervention in a new drug makes them more than minimal risk. New surgical procedures, new medical devices, any of those clinical trials and interventional studies would get reviewed by that full convened board. Now, a lot of researchers have the impression that they submit protocols to the IRB. The IRB goes off into a closed room and they talk about some ethical stuff and it's a big black box, and then they come out with a decision that may or may not make sense. And they give that result back to the researchers and tell them whether they can continue, but there are actually specific criteria for whether the IRB can review a research proposal, whether they can approve it, that are based on the regulations. And those criteria are that the risks of the research have been minimized as much as possible, that the risks of the research are reasonable in relation to the anticipated benefits from the study. And that includes both direct benefits, so potential benefits to the people who are enrolled in the study, that it might help delay the progression of their cancer, that it might help their quality of life, but also benefits to the society in general. So is the research designed in a way that will answer this research question so that it will move the study forward? It will move the progress in this area forward. So even if a study doesn't have the results that we hope to see, even if it's a negative study in that the new drug didn't work, if the study was still conducted in a rigorous way so that it answered an important research question, that still moved our field of treatment forward. Even if that new drug didn't work, it showed us that we need to put our research resources into a different area. There have to be provisions for the monitoring of data and the safety of the participants. Informed consent has to be obtained and has to be appropriately documented. And there have to be precautions for the privacy of participants and the confidentiality of their data, and that the selection of participants will be equitable. And then also, as I mentioned, if we have vulnerable populations who are either vulnerable to undue influence about whether to be in the study or are decisionally impaired in some way. So people who have advanced Alzheimer's disease or people with other neurological diseases or either temporary or permanent conditions that impairs their ability to make decisions may need additional precautions, like someone else to provide consent on their behalf or something like that. So these criteria all track right back to those principles of the Belmont Report in terms of respecting autonomy, ensuring that there is distributive justice in how we select participants in the study, and trying to do good in our studies. Informed consent is, of course, an essential component of research. And we often hear people review to the informed consent document or the informed consent form where they say, I got consent for the study, meaning they got someone to sign a form. But it's important to remember that informed consent is not a document. It's not a piece of paper. There is a piece of paper. They're way too long, but there are pieces of paper. There are documents that we give people who participate in research that basically provide a reference document for them to take home with them, that gives them all the information about the clinical trial and what's going to happen in the study. But that shouldn't be the entire consent process. Informed consent is a conversation. It's an interactive conversation and a process, and it is continual through the entire course of the study, as evidenced by the fact that anyone can withdraw from a clinical trial whenever they want to for any reason. The basic elements of that consent process are that there has to be full disclosure of the nature of the research and what it means to participate. There has to be adequate comprehension of that information on the part of the potential participant, and they have to be able to ask whatever questions they want to, and they have to make a voluntary choice to participate. So that's a very quick and very brief description of how research participants are being protected. I know we don't have time for questions today, but I'm going to put my email address in the chat box. And if you have any further questions or comments or feedback, anything else you'd like to tell me, you're welcome to reach out to me directly. And I'll hand it back over to the organizers.
That was a fantastic behind the scenes of all of the different elements that we think about when we're thinking about it. But I think that the theme that I think one of our moderators, who's one of our residents, mentioned is the informed consent is not a form, the process. That is like a really critical thing for all of us as clinicians, researchers, and as patients to think about. So thank you.
Yes, thank you, Dr. McNair. That was an excellent transition into the topic I'm going to cover. My name is Joanna Burdette, and it's my absolute delight to be at this third annual meeting. I'm a basic science researcher at the University of Illinois, Chicago, and I'd like to talk a little bit about how important patient tissue and samples are to research and making new discoveries to expand our options in prevention, therapy, and safety that we see in gynecologic cancers and other cancers as well. Next slide, please. Yeah, go ahead. So one of the types of research that I am involved with a lot is the use of patient samples. And that is because although we can make a lot of important discoveries in terms of new molecular targets and strategies, we need to study human beings. And it's very important because each and every tumor is unique, and we want personalized medicine in order to have a more robust response and to have better options for patients. And so, as was just so elegantly described, was the process of asking patients to be involved in research by donating human samples and all of the rigorous inputs that go into that. But there's another side to it as well. And that is the side of the researchers using these samples in order to investigate them to find new ways of understanding the disease so that we have better options. And so there's definitely a human cost to this, and there's an economic cost in terms of collecting the samples, curating them, making sure that they are de-identified so that privacy is protected, and that we know enough about the sample so that we can make meaningful interpretations on it. Is this really high-grade serous cancer? Is it truly endometrioid? Are we actually looking at what we think we're looking at? And so there's an obligation on our side to use these samples in an ethical and to really use them for as much as we can to try to understand as much about these diseases as we can. Next slide, please. So there's a few ways that scientists think about using these samples. One is retrospectively, so looking backwards. So one of the things that's important is to collect enough diversity of patient samples in order to make actual understanding about our disease in a way that many patients would respond to a therapy, not just one or two. And also to understand which patient populations a drug could be used in, or a new device could be tested in, or an imaging modality could be informative for. And so we really need a lot of patient samples in order to understand that. Another way to think about this, though, would be looking forward. So once you have a certain diversity of patient samples, you can start to make hypotheses about how a tumor might respond to a new idea, a new surgical intervention, a new therapy, a new imaging modality. And that can really give us new options in the toolbox for mitigating these diseases. And so one of the most important ways we think about this is cryopreserving or storing these tissues so that there's enough of them and they have enough diversity in them to actually make conclusions. But another is actually growing and studying them as live cultures in the laboratory. And I'll talk a little bit about that going forward. Next slide, please. So there's a lot of different samples that researchers use, and they can provide different kinds of information. And we often, when a lot of people think about being involved in research, they might think about a clinical trial. But sometimes being involved in research is simply saying, this tissue you are already going to have removed for your disease, would you be willing to donate it as a de-identified sample to a researcher? And so it doesn't mean you essentially need to just agree if you're okay with that or not. And some of those samples that might be requested could be follow-up samples like saliva, blood. They could be the tissue core from the biopsy itself. Those tissues could be stored. They could be frozen for RNA and protein. They could be formalin fixed, so we can look at histology and different markers with spatial information. They could be fresh tissue that could ultimately be grown and understand, is this tumor aggressive? Does it spread? Where does it go to? How could we mitigate that? And then there could be samples that are collected in primary treatment, follow-up, and those give us different understandings of disease, how well a patient responded to a therapy, why didn't they respond, what's happening now, how can we treat that disease as it emerges and evolves over time. Next slide, please. So one thing that came up in our breakout session is it's important for people involved in this to understand the success stories so that they will so that they'll want to be involved and they'll see the reason behind it. So a great success story is the Cooperative Human Tissue Network. This is a collaborative group that has banked and stored and distributes millions of human biospecimens to investigators so that they can actually understand disease. And this has resulted in thousands of publications that have really transformed our understanding of human diseases. And so two really powerful examples would be the molecular targets for breast cancer that we think of, estrogen receptor, progesterone receptor, and the HER2 receptor, which there are targeted therapies for that are very useful and have prolonged lives. And in ovarian cancer and high-grade serous ovarian cancer and other types of cancers, BRCA and homologous repair deficiencies and in tissues have now informed physicians of using different unique therapies like PARP inhibitor therapy in maintenance therapy and in recurrence. So this is really important. Without these targets, we wouldn't have understood to use these drugs in these patient populations, and understanding more about the tumor allows us to expand the patients that are allowed to be treated with these drugs. Next slide, please. And something that you might not think about are tissue engineering. So this is really important if we want to understand prevention, because when we engineer in the laboratory cancers, we can start to put into the model system things that prevent the cancer in the first place. So this is really important in cancers where obviously there's poor diagnostics. We really want to tackle this from a way that would affect major change in millions of women. Prevention is a huge way to do that. And so it's really interesting to think about that. The other thing we want to think about a lot is the idea that tumors are not just cancer cells. We've learned more and more. They're the endothelial cells, the blood vessels. We can have drugs that target those. They're the immune cells. We can have drugs that target that. They can be the surrounding extracellular matrix. They can be the adipocytes, the fat cells. They can be the stromal cells. All of those things, if we can understand their role, provide unique opportunities to actually slow the growth of cancers, diagnose them better, prevent them, et cetera. So these engineering approaches using real human samples really open up a lot of opportunities in the laboratory. Next slide, please. So thank you so much for giving me the opportunity. In an era of COVID, we've actually noticed quite a few patients deciding not to donate tissue. And we understand that there's a lot of issues right now, but as a researcher, it's vital for us to understand each and every tumor and really have access to patient samples in order to make change. And so we appreciate your consideration of the donation and the opportunity to speak today. Thank you so much. So we'll move to a stretch, and Gina Curry, I believe you're going to lead us in this.
What I think between Gina and myself, we will do
Dr.
this.
Burdette. Okay, yeah, I was going to say, actually, it's going to be just a video, and so you can take this time. And Dr. Lee, I didn't mean to cut you off. We're going to take this time to show the video, and then, but if you want to, take a break and go to the restroom or whatever you need to do, feel free. You don't have to necessarily watch it, but it's always a really great video to show, especially at this the type of theme this is. Go ahead, Dr. Lee. I'm sorry to cut you off.
Oh, no, thank you. Thanks everyone for your attention and this fantastic presentations. I love Dr. Burdette. You totally rock in terms of being such a model for this. So here's our break. Take a break for 5 minutes. I'm going to share screen. And hopefully this will work because I want to make sure I'm doing this correctly. You might see my messy desktop in the background, but we will be starting our exercise to kind of think through. Ooh. Let's see if it's working. All right, here we go.
Let's make it a little bigger if you can.
Okay.
Okay, first we start with our
You know, full screen maybe.
cancer prevention.
Perfect.
Cancer prevention. We usually do 3 of them. Cancer prevention. Our first pose is the parent pose. Safe and effective. HPV vaccination is safe and effective. Third time like you mean it. Safe and effective. Pose number 2, partner pose. HPV round table. HPV round table. One more. HPV or your local state round table. Third pose is the provider pose. So some of you know this is warrior pose. Left foot forward, right foot back. One arm forward. Vaccination. Finger ready. Shot 1. Shot 2. Over age 15, you need a shot 3 and vaccinate. Fourth pose is patient pose. After getting the vaccination, you sit and wait 15 minutes so patients don't faint. They stay in the office. Feel the burn? Yeah, we're doing 3 of these. Take a little break and patient pose. We're waiting. Only 10 minutes left. We don't want those kids to fall out of their chairs. This is how we keep our kids safe. And one more patient pose. And now we are fully vaccinated. Thank you for joining us for HPV Champions Yoga.
Namaste.
Just our little shout out for GYN cancer prevention in terms of cervical, vulvar, vaginal prevention with HPV vaccine. So I know it's much more fun in person. So maybe next year we'll do it in person.
All right, I'd like to transition us into the portion of the day for the research presentations and just a little background. So Kimberly Richardson and several MD PhD students at UIC, including a student in my laboratory, Tova Bergston, were involved in a program called, which is still ongoing, called Survivors Advising Scientists Education Program, or SASIP. And the goal here was really to have more learning between survivors and scientists, so that there were better educational tools and that there was integration into medical curriculum about interacting with survivors and patient advocates and caregivers. A facet of this, which maybe we haven't discussed much today, is actually the review of scientific proposals and reading scientific publications, which have a lot of breakthroughs and new information that can really be empowering to our community. And so today we will explore that interaction by listening to scientific presentations. So thank you so much for being here.
Okay, we're going to start. Basically, these are our junior researchers, if you will, for lack of better terminology. We're going to start with Nada.
Thank you so much for having me today. Let me just pull up my slides here. Okay, can you all see that? Perfect, thank you so much. So my study was called the Current Practice Patterns for HPV Vaccination in AYA Cancer Survivors. So what a lovely transition from yoga to my presentation. So a little bit of background, what is AYA cancer survivors? So this is a group of patients that have been diagnosed with any type of cancer between the ages of 15 and 39. This group is unique because they have an up to 3 times increased risk for developing a second cancer over their lifetime. And some studies have even showed that female survivors have a 40% increased risk of developing an HPV-associated cancer in their lifetime. So it makes perfect sense that this group that is at increased risk for developing a second cancer would get the only FDA-approved cancer preventing vaccine, which is known to prevent 5 types of cancer, including cervical, vaginal, and vulvar cancer. Unfortunately, the rates of vaccination in this group are still very low, even though HPV vaccination is recommended to cancer survivors in the National Cancer Comprehensive Cancer Center guidelines. So my study was looking at whether or not the HPV vaccine is actually being recommended to AYA survivors. And we did this by surveying clinical oncology providers across the United States through the American Society of Clinical Oncology. We surveyed any type of clinical oncology providers, whether that be doctors, PAs, or advanced practice nurses that are in active practice and care for AYA survivors. We received 217 survey responses nationally it was pretty diverse in regarding Northeast, Midwest, the South, the Western part of the United States, and also pretty mixed in terms of geographic location from rural, suburban, or urban centers. 77% of our respondents said that they do care for AYA cancer survivors. Our primary endpoint was looking at age-appropriate HPV vaccination for those less than the age of 26, and only about 42% of our respondents said that they recommend the HPV vaccine for this group. In 2018, the FDA actually extended the age range for the HPV vaccine to the age of 45. And unfortunately, only 10% of our respondents said that they recommend the HPV vaccine to those less than the age of 45. I wanted to include some of the comments that we received, and just for the sake of time, I won't read them, but a majority of the comments received from our survey respondents were that this is something that is extremely overlooked and kind of honestly not on the radar of most of oncologists. A lot of the oncologists said that they're more focused on treatment, but they are increasingly should be involved in the role of prevention as well. So in conclusion, only less than half of the oncology providers that we surveyed recommended the HPV vaccine to those less than the age of 26, and even more so than the age of less than 45. We found that providers were more likely to recommend the HPV vaccine if they're familiar with the national guidelines that are in place. And we didn't find any differences in vaccination recommendations by whether or not you are in a specific geographic location, your population type that you served, rural, suburban, or urban, or whether or not you are like a medical oncologist, gynecologic oncologist, or pediatric oncologist. And then lastly, hopefully we can just spread the word out there and make these guidelines for the specific role of the oncologist in cancer prevention, not just treatment, and also kind of fine tune when the optimal timing of receiving a vaccine in relation to chemotherapy and radiation treatment, because those specific guidelines currently do not exist. Thank you so much for allowing me to present this.
Thank you, Nada. And we're going to transition now to Andres for his presentation.
All right, thank you so much for the invitation. My presentation, let me share the screen here, is a little bit more in the basic science. I am a basic scientist, and it's titled Understanding the Interaction Between Ovarian Cancer and Fat. So you may be wondering why it's important to understand this interaction, why these two cells are actually important to understand. So it's widely accepted now that ovarian cancer originates here in the fallopian tube and then is able to migrate into the ovary. And in later stages, cells from these sites are able to move into the peritoneal cavity and then attach to this fat layer here that protects our internal organs, that is called the omentum. Here, cells are able to spread to other sites in the omentum or detach again to spread to the fallopian tube again or the other ovary, creating further metastasis sites. So the cancer cells during the process of metastasis are in close proximity and in close contact with fat cells. So in order to understand the interaction between these two cells, in our lab, we set up to generate a model in which we can replicate the process of metastasis in a dish. And for this purpose, we sit fat cells on a dish, we differentiate them for 7 days, maturing them so they can actually acquire and accumulate lipids inside of them. After that, we can use cancer cells that we label with a green fluorescent on top of them, just like if they will be invading in the body. We could culture them for 3 days, and then using a machine, we're able to separate them and study these cancer cells that were in contact with the fat. Here's an example of our co-cultures. You will see here, this is the fat cells. We can recognize them because the cells accumulate fat. These bright spots inside of them are the fats. Here in green, you will see the cancer cells, and here you will see the two cells growing together in a co-culture. Furthermore, we can actually label now in green the fat cells, and in red are the cancer cells, and we can reconstruct in 3D this model. And as you can see here, cancer cells are able to grow on top, besides, and even sometimes underneath the fat layer, closely replicating what they will be doing inside your body, but in this case, in an in vitro dish. So what happened with the cancer cells when in contact with fat? Well, we found out that cancer cells proliferated faster. You'll see here the cells that were in contact with fat grow faster than the cells that were growing alone. They become more resistant to platinum, in this case, to drugs. In this case, the ID50, that is the minimal amount to kill half of the population of cells, went from 7.54 micromolar to 9.32 micromolar. And they also become more aggressive and invasive. As you can see here, the cancer cells that were in contact with the fat were able to migrate more through a porous layer in this case. So the great question here is, what is happening with the cancer cells that are in contact with fat? We know that fat accumulate lipids that can go inside of the cancer cell and generate a cellular response, but we don't know what's the effect. So we take these cancer cells and we sequence the genome, and we found that two proteins that belong to the immune system were actually increased in the cells that were in contact with fat. In this case, 13 times and almost 2 times more. So we then study what are the pathways that are activated in the cancer cells that were in contact with fat. And once again, we found out that the activation of C3 and C5 was important. And not only that, but the cells that were in contact with fat, these cells were able to generate more C3 inside of them. So C3 was shaping up to be one of the responsibles, was one of the proteins responsible for the increased proliferation. But in order to prove this, we had to block C3. In this case, we use a chemical inhibition of C3. Here are the cells that grow in contact with fat. This is the cells that grow alone. And here are the cells that we treated with the inhibitor. And as you can see here, we were able to inhibit the growing of the cells. We then use a genetic reduction of C3. We block the production of C3 of the cancer cells. And when we did that, we see exactly the same results. And as you can see here, the cells that don't have C3 inside of them grow less than the control cells and the cells that were in contact with fat, proving that C3 may be the main effector that drive the increased proliferation of cells when they were in contact with the fat. So the conclusion and future direction of my work, we established a reproducible, direct, and long-term co-culture system for the study of interaction of cancer cell and adipocytes. This system can be used to test phenotype of cancer cells in animal models in the future. The co-culture system could be used to establish more complex models using other cell types like endothelial cells, fibroblasts, and other cell types. And the study of the interaction of cancer cells and adipocytes could allow the development of new drugs in the future to stop the metastasis of cancer cells. Finally, I would like to acknowledge the laboratory of Dr. Daniela Matei with all the previous and current members, Dr. Julie Kim, Northwestern University, and Dr. Elias Kinesius for the University of Iowa for providing the fat cells, and all of you for being here and paying attention to my presentation. Thank you so much.
Thank you, Andres. Thank you so very much. And now we will move to Raul.
Let me just share my screen here. Can everyone see the screen okay?
Yes.
Well, good morning, everyone, and thank you so much for giving me the opportunity to join you all and share some of the exciting work that we're doing. My name is Raul Krishna, and I'm a GYN Oncology Fellow at the University of Chicago. So I finished my ObGyn residency, and now I'm in year 2 of 3 now of my training to become a GYN oncologist, much like Dr. Lee, Dr. Kernan, Dr. Yamada from our group here, and so many others I want to share a little bit about the research that I've been working on this past year, which is looking at one subtype of ovarian cancer called low-grade ovarian cancer. We'll talk a little bit about that. I have the good pleasure of working with a very talented group of mentors here at the University of Chicago. As you can see here, Dr. Lees are, in addition to all the things she does, she's also the fellowship director and my direct mentor in training as well. This is the Ovarian Cancer Lab at the University of Chicago. Dr. Ernst Langiel, who's our department chair, is also, he runs the Ovarian Cancer Lab, which has been very successful over the last 20 years or so in doing some very exciting research. So I've had, in addition to working in the hospital, the good privilege of also being able to work over the past year in the lab itself. And I think part of the reason we this is done is so that future clinicians who are going to take care of patients also engage in research of some kind. It could be basic science research like I've been involved in. It could be clinical trials research. It could be patient experience research. Want to make sure that the physicians who are going out there and taking care of patients are also advancing the field. So patients 5 years, 10 years from now have a even better experience and outcomes than we're able to offer today. I just want to take a step back and just quickly go over low-grade ovarian cancer. So what is it exactly? So when we think of ovarian cancer, most patients who are affected by ovarian cancer have what's called high-grade ovarian cancer. That's typically, when we say ovarian cancer, what people think about, but low-grade ovarian cancer affects still a very significant number of women every year. It's a less common subtype compared to high-grade ovarian cancer. You can see here, this is the uterus in the middle, and both ovaries are affected by this tumor that you can see, and that's the actual low-grade tumor here. The reason we call it low-grade is that the tumor cells in these cancers are much slower growing than the ones in high-grade, hence the term low-grade. They usually affect women at a much younger age, so ages 45 to 55, whereas for high-grade, we typically think of these at a slightly later age, around age 60, although there's quite a bit of variation in that as well. Low-grade ovarian cancer still presents in a very similar way to other ovarian cancers. So often patients will come in complaining of bloating, abdominal discomfort, changes in bowel function. What makes low-grade ovarian cancer especially frustrating, though, is that it doesn't respond well at all to the traditional chemotherapy. Things like carboplatin and Taxol, these chemotherapy agents are good at killing fast-growing cells, but as you can imagine, low-grade ovarian cancer cells, because they grow much slower, aren't as sensitive to these chemotherapy agents, and this can make it very hard to help our patients. And ultimately, because it's a rarer type of ovarian cancer, it's also not as well studied compared to the more common high-grade ovarian cancer. And so really, there's a paucity of good research out there, and makes it hard to know how to help our patients. What's interesting is that low-grade ovarian cancers, they're thought to arise from a precursor tumor called a borderline ovarian tumor. And these don't necessarily act like true cancers, but there's something that happens that causes these pseudo benign tumors to become invasive malignant cancers and spread and affect our patients. So the question we sought to answer was, what are the changes, both in the protein and RNA, that occur that cause these low-grade cancers to develop and spread? I want to introduce quickly a concept of spatial biology, which is something that'll become important to discuss here. When we think about here, what we see is a microscope slide of an actual low-grade cancer of a patient's ovary. What you can see on the left here in the red circles is the low-grade tumor in this dark purple outline here. But on the right, you can see also a concurrent borderline tumor. Now, previously, what would be done in research is you would take a tumor sample like this and process it all together. The problem here is that we lose details. We lose the ability to differentiate what makes the tumor on the left different from the tumor on the right. And the other thing is, as some of our previous speakers have said, it's not just the tumor cells themselves, it's the other tissues that help support the cancers as well. So in the light pink you see here is the stroma. These include the immune cells that can fight or somehow not fighting these invaded cells, blood vessels that feed these tumors, the stroma that helps support it. We lose a lot of that context. So spatial biology basically tries to separate these two so that we can understand what makes each site. So we used two novel spatial technologies to study this cancer. The first was something called spatial proteomics, where we study the proteins that make each tumor unique. And then we use something called spatial RNA to study how these tumors express their genes as well, mutations, and what are the consequences of that. You can see here that, at the bottom, we actually take tumor samples from our patients who give us permission, and actually can then make microscope slides and actually study it in this, which is an incredibly powerful technique, because now we can look at patients from decades ago and go back and actually look to see what is it that made their tumors unique. The first methodology is one that we actually were collaborators with the Max Planck Institute in Germany. It's a very new technology that that was just published this year in Nature Biotechnology. It uses laser microscopy to actually cut out individual cells, and we can then study the proteins in this. This image here shows you a microscope slide of a fallopian tube, and you can see here that using stains, we can actually now separate the two different cell types that are present here and actually look at what makes each one unique and study those. This is an actual live image or video that actually shows you how we can individually cut out tumor cells. So we're not getting contamination with non-cancer cell types, other cancer cell types. You can get an actual understanding of what the profile is of each cancer. So similarly, just like we did with proteins, we were also able to study RNA. So how do cells use the genes that they do? And how does that help their functioning? So here you can see on the left is another microscope slide. The dark purple here cells are the tumor cells. And we can actually separate the tumor cells from the non-tumor cells to get a pure understanding of this. So over the past year, we implemented these two new methods to about 16 different patients who had low-grade and borderline tumors. And we've been able to characterize over 7,000 genes and 6,000 proteins in these tumors. What I want to quickly show here is on the left here, you can see these are genes and proteins that are very high in low-grade cancers, but not in the borderline tumors. And on the left, we can see genes and proteins that are very high in the borderline tumors, but not in the low-grade tumors. And trying to understand this helps us really understand what is it that makes borderline tumors progress to low-grade tumors, and what is it that makes low-grade tumors able to spread and become so dangerous to patients. Now, the hard work really begins at this point, because we have all these wonderful targets and promising genes that we can look at. Our next step is to try this, to actually test this out. So we can either suppress or inhibit these targets in low-grade cancer cells, or actually try these in mouse models as well to see which of these are actually promising genes targets. But it's a wonderful data set that we can use, and we're planning to share with the research community so that others also can access it, and we can attack this on many different fronts as well. I can't emphasize this enough. It's, projects such as this require a lot of collaboration. So I want to thank Dr. Ernst Langell for his mentorship as well over the last year, as well as our collaborators in many different fields. So in Germany at the Max Planck Institute, where they developed a lot of these technologies, our pathologists, our genomics folks, our bioinformaticians who help us analyze this. And lastly, this can't be said enough. It's our patients and their families. I think Dr. Burdette talked a little bit about the tissue collection process. It always amazes me how when you meet a patient before their surgery and you ask them for permission, you can only imagine the anxiety and stress that they're going through. And yet, when a patient says, you know what, I'm happy to do what I can. This may not help me today, but if it can help someone else in the future, I'm happy to do what I can. And it's a very humbling thing when you see that. So just want to give a very special thanks to our patients. And thank you for giving me the opportunity to talk to everybody.
Thank you, Raul. And now we're going to do our breakout session, our last breakout session. This one will only be 10 minutes because we need to recoup a few minutes of time. But here are some of the things that we are going to try to get an opportunity to suggest and have a discussion around. As we talk more about how we're transitioning from doing educational modules between patient advocates and junior researchers, we're taking that same framework into this discussion in the breakout rooms. So we want to ask you, what did you as a listener understand to be the goal or the purpose of the research you heard today? How would you describe this research project to a friend after hearing about it? Do you have any feedback for the presenter on how they could have made their research work more understandable to you or to patients or to caregivers? And then lastly, what else would you like to learn about their research? Now, don't worry, all these questions, you don't have to memorize. Your moderators have them. And so we're going to ask you to go into a breakout room where you're going to be in with one of the researchers that just presented. And then we're going to come back in less than, maybe a little under 10 minutes and finish up with the rest of the summit. So we'll take you into breakout rooms now. Thank you, presenters. We appreciate you.
Thanks everyone for this great overview, for the great presenters, as well as everyone who is providing feedback and thinking about how best we can engage. I think we can do the next slide. We want to do a quick tour of behind the scenes of how GYN cancer research projects are developed. Each researcher and presenter is going to go over a quick slide and go over their a little bit of their background, and we're going to try to shorten it a little bit because we know time is of the essence and we're running over with so many good topics. I would like to really emphasize here at this panel, when you're looking at this, how important it is that we see people who are clinicians, people who are patients as well in the researcher PI space. And I think this is just really great to see. So I'm going to go ahead and have Dr. Kernett start. Or no, here we go. All right, excellent.
Hi, thank you. Can you click once? Yeah, I think that there may be animations accidentally. There we go. So hi, thanks everyone for letting me talk again. I'm just going to give a brief overview of some of the work that I'm interested in. There's kind of lots of different types of research like we talked about. And this project that I'm working on is more on the translational side. So Dr. Krishna just talked to us about some like translational type projects. This is a similar kind of overarching theme, but this is specific for a type of endometrial cancer called uterine carcinosarcoma that I'm interested in, I'm trying to further study. So I guess I'll start at the bottom with the why am I interested in this? My interest started with kind of broadly endometrial cancer. I'm really interested in biomarkers and how do we help find new treatments, but also help predict which patients are going to do really well and which patients are going to need additional, help down the way. And specifically for uterine carcinosarcoma, this is a rarer type. So even though it is less common, it tends to be pretty aggressive. And I put a little schema on the side that I took from a paper, but it basically is a more heterogeneous, like there are different parts of this tumor that just make it really interesting to me from like a scientific and biology perspective. And I think that this lends itself well to some really creative and unique ways that we can approach treatments. But from like a bigger societal picture, I know that for endometrial cancer, mortality is one of the few cancers that we're actually seeing it rise, which is really appalling in the kind of state that we're in with cancer care. And especially these disparities that we're now seeing, especially in endometrial cancer, where we're seeing African-American and Black women having worse outcomes, having higher mortality rates. And that's something that I know that we need to not only address and fix, we need to understand why. And for me, part of that is understanding some of these rare endometrial cancers that are much harder to treat. And so for this study, what we're doing is, getting patient consent, offering enrollment on the study, where we collect tumor pieces when we're doing surgery that they would have had anyways. And then I'm doing some studies in the laboratory to test the tumor for certain types of proteins and DNA changes and kind of effects, so that overall, kind of what I'm interested specifically is looking at how the immune system interacts with these cancer cells to try to find new immunotherapies or newer types of treatments that might be able to help patients with this type of cancer. So this involves patient tumors, but also involves cell lines or kind of cells that have been developed over years, or even like mouse models where we can help test treatments that may be helpful for patients, but before we bring it to the patient side. So I'm excited if anyone has any interest or wants to talk more. I always love talking about research, so please feel free to reach out.
Thank you so much.
All right. Hi, everybody. I'm Dario Roque. I'm one of the GYN oncologists at Northwestern. I've gotten to meet a few of you on the small groups, but I'm really excited to be here. And I was also really thrilled to be part of the organizing committee. And thank you, Kimberly and Dr. Lee. You really have done a wonderful job, and I can't wait to participate in future years. But this is a very brief overview of a project that I'm in the process of getting started at Northwestern. So I am very interested in the role of what's called HIPEC, which stands for heated intraperitoneal chemotherapy in the treatment of ovarian cancer. So currently, that is a treatment that we're only using in patients that have stage 3 and that are going through an interval debulking surgery. So this is a very small subset of the women that are undergoing treatment for ovarian cancer. And my primary goal was to try to understand better the biology that happens after women are treated with HIPEC. More specifically, I wanted to know, does the changes that happen after HIPEC, we do know that they seem to lead to better what's called a platinum-free interval. And for those of you who have gone through ovarian cancer, you know that that is like a very meaningful definition of kind of like what we use to choose therapeutic regimens. So we always talk about platinum-resistant disease or platinum non-resistant or platinum-sensitive in a way. And in a way, we know that that platinum resistance or platinum sensitivity can be correlated not only with response to platinum, but also what's called like disease-free survival. We know that it can also be correlated with what's called like homologous recombination deficiency, which is a test that we are routinely doing in women with ovarian cancer these days. And that women that are homologous recombination deficiency, we know that they tend to respond better to PARP inhibitors. So there's a lot of factors that go into the treatment decision. So what I'm trying to do with this project is being able to collect tissue right before, and this is all done at the time of the surgery, but right before the patients are treated with HIPEC, and then leave a small piece of cancer inside the body during the HIPEC treatment, and then we would remove that piece of tissue right before finishing the surgery. So essentially, we're still removing all the cancer, so we're not doing anything differently than what we're doing, but by leaving like a small piece of cancer during the treatment with the HIPEC, then we can compare that piece of tissue with the main cancer before treatment with the HIPEC and see how those molecular changes can help predict either the platinum-free interval or potentially be a better marker than homologous recombination deficiency itself. So this is very exploratory in nature. We're only planning to recruit about 20 patients or so for this study, but it is more like a translational rather than an interventional type of study, because if you think back to Dr. Kerner's earlier talk, we're not changing what we're doing. We're just trying to get more information in terms of tumor biology. And then I really want to acknowledge Friends of Prentice for funding this project. And as I'm saying, we're in the process of getting it started. It's being submitted through the IRB. It's a very important part of this research. And I'm happy to take any questions now or in the future. Feel free to reach out to me via email. My Twitter handle is listed there. And again, thank you all for being here and supporting this event.
Thank you so much, Dr. Roque.
Hi, everyone. Excuse me, Shannon McLaughlin David here. Good to see you again. This project that we are in the process of writing this protocol at the University of Illinois is ashwagandha and cervical cancer treatment. It's a clinical and translational trial. And ever since Dr. Kernett gave her talk earlier this morning, I've been trying to figure out which box to put it into. And it really has a little bit of all of the components. Possibly because evidence-based medicine, as we have described it here today, is really like a 20th century thing that was described and defined for the first time. But medicine is as old as humanity. And so we're still a fledgling, clinical research is still a fledgling thing. And I bring that up because this project is focused on a concept that we have all heard from someone we loved, which is stress is bad for you. And we are learning on the molecular level just how bad stress is for cancer. And this project is focused on our patients with cervical cancer, which is, as I mentioned earlier, it should be a completely preventable disease, but we are not succeeding in prevention, and stress and cervical cancer go together. So we know HPV causes cervical cancer. We know that stress on the body helps the virus do what it wants to do and helps the cancer once the cancer is formed. Cervical cancer as a disease is associated with a high allostatic load. Allostatic load is kind of a fancy term for different kinds of stress coming from the outside or chronic illness within our own body. And ashwagandha is an herbal supplement available over the counter. It's been around for about 600 years, or at least the documentation of it is as old as Ayurvedic medicine, which is about 600 years old. And it actually has 20th and 21st century data supporting that ashwagandha as a supplement brings calm under stress. So this study is implementing ashwagandha on top of whatever cervical cancer therapy that a patient is being prescribed, whatever stage they are at the time of their diagnosis, so that we think ashwagandha will help reduce stress. But we also know, at least in petri dishes and mice who don't have immune systems, that widoférin A, which is a molecule within ashwagandha, that widoférin A actually inhibits the proteins from the HPV virus that cause the cancer and has actually shown some preclinical data that it will shrink cancers. So we're hoping this is a 1-2 punch for our patients with cervical cancer. And I will leave it there in the interest of time.
Hi, I'm Dr. Barney McGregor, and I want to thank Dr. McLaughlin for introducing the concept of stress. And I want to thank Dr. Krishman for talking about tumor biology and how the immune cells can get into the tumor microenvironment. I'm a clinical psychologist, and I work with Dr. Susan Leckendorf at the University of Iowa. She spent years studying the effects of chronic stress on immune function in the tumor microenvironment and ovarian cancer cells. And she's shown that chronic stress can have result in decrements in immune function in peripheral blood, in the ascites, and in the tumor microenvironment. And guess where the effects of chronic stress are most potent? Right in the tumor microenvironment. She's also shown that social isolation can be associated with poor prognosis after ovarian cancer treatment. It got to the point that she really felt like she needed to do something. Like Dr. McLaughlin's got the ashwagandha to try and make a difference. We want to make a difference too, but what we're doing is we're offering a 10-week cognitive behavioral stress management and coping intervention. It's got a lot of mindfulness practices in it. She came to me to work on this project because I had done research in breast cancer patients showing that this 10-week talk therapy intervention, it's like a series of workshops, could improve not just mood, but also immune function in early stage breast cancer patients. And more than that, we showed that this talk intervention, just working on things in your head, could actually change which genes are turned up and turned down in response to adversity. So we can show that this talk therapy actually has changes at the level of the mRNA, like Dr. Krishman was showing. I'll tell you a little bit more about this. My whole reason for being here is to let you all know that this is available. It's a national study. It's for women who have been diagnosed and treated for ovarian cancer. If you're within 5 years of the end of your active treatment, you are eligible for this study. You can be anywhere in the United States. We even have somebody in the Caribbean and I think people in Canada who are participating. So if you sign up for the study, you can go to livingwellstudy.org to find out more about it. You'll be randomized, or we flip a coin to see which one of two conditions you get. One is a 10-week mindful and stress management training program. The other is healthy lifestyles. We interviewed ovarian cancer survivors to see what health concepts they wanted to learn more about, because we wanted to make sure that everything that we were providing for ovarian cancer survivors was something that they wanted and needed. So we'll either get stress management and coping techniques. We know ovarian cancer is very stressful. It can be isolating. We've had women in the groups that have never met another ovarian cancer survivor. And we also know that women want to know more about exercise and supplement use and sexual functioning after cancer. And by the way, what do I do about my difficulties with sleep? So in the Healthy Lifestyles program, we've got 10 different experts in all these areas who will give you a 30-minute lecture, and then there's a lecture on all these different topics that are of interest to ovarian cancer survivors. I'm not sure how much time I have left, but it's a really great program. We've had women say this program was a game changer. We've had women say, I didn't know that I needed this, but I did. It's hard to describe what this is. We've been having people try to help us describe it. One woman said, this is the best thing that I've done since I was diagnosed. You meet in groups of 6 to 8 women. The groups are about 2 hours long. You meet via Zoom video conferencing. It's so funny, when we first started the study, we used to have to show people a little screenshot. This is what Zoom conferencing looks like. We don't have to do that anymore. This started up right at the beginning of the pandemic. We felt very, very smart that we already knew how to do this. If you don't have a laptop or an iPad, we will send you a tablet that has cellular data access, so you don't even have to have Wi-Fi in your house. So just go to livingwellstudy.org and you can read more about it. There's also a chance there for you to log in and do a screening survey to see if you're eligible, and then somebody will contact you. We've shown that this can improve, these sort of interventions can improve survival in breast cancer patients, and we think that we can do the same with ovarian cancer patients. Plus, we just know ovarian cancer is hard, and just having this support we think is really important. Thanks a lot for listening, and I hope that I see some of you in the groups down the road. Thanks a lot.
Fantastic. Thank you so much.
Okay, well, I'm Amanda Leong, and I am representing Vogue, Black Voices on Gynecologic Cancer Understanding Experiences. And to let you know, I was actually a survivor speaking at this conference not that long ago, and now I run a research project that I started, which can be done by anybody. If I can do it, anybody can, really, seriously. I sent one tweet on January 3rd of 2021 that said, I'm going to focus on racial disparities in gynecologic cancer. And that was because we see the research, and we know through our friends and loved ones who are Black and Brown experiencing the same cancers that we are, that this is a problem, right? We know racial disparities are a problem in gynecologic cancer. And so for me, knowing that cancer is already so challenging, it's just like, goodness, we got to do the most we can to try to work on this. So Dr. Uni Balagon responded to my one tweet that maybe had 3 likes. It went nowhere, except it went to one right person. And she DM'd me, we got together, we created an incredible team. Our team is literally made up of patients and caregivers like Candace Draw, who's on this call here. She's there. It's wonderful because the absolute GOAT of patient advocacy, Kimberly Richardson, is also part of this amazing work. And so we learn from her all the time. She's probably why this exists, because I got inspired by her. And so basically what we're focused on is, we know that this quantitative research is out there, but why has nobody asked the Black patients who are actually experiencing the different outcomes and their cancer care differential. Why isn't anybody asking them, like, okay, tell us about your cancer care so that we can like hear in your own voices what is happening, right? And so qualitative research is not always valued very highly in medical science, we know. Neither is patient-included research always, but we're just determined to make this something that other people can learn from and replicate, because we're actually having these focus groups of Black patients with gynecologic cancer, and then we're listening to them, and we want to amplify what we learn in their voices. And in order to have solutions that are going to be guided literally by the people who have the worst outcomes on these cancers. And so it's absolutely challenging. You can't imagine having all these amazing physicians, and we've got physicians, we've got epidemiologists, we've got social workers, we've got nurses, we've got a lot of, some amazing Black patients on our team who lead the way. And the challenge we have is to center the voices of Black patients. And it's a really fun challenge too, because it's a lot of passionate people. We're funded by Cornell Center for Health Equity and get a lot of great support there. And so what we're really doing next is we're building on our first focus groups. We're going to do 10 more focus groups coming up in probably like the winter time. And then we're going to get some better descriptions of our survivor sample. And we're going to be holding some healthcare stakeholder sessions that I know Dr. Nita Lee and some other of our wonderful people who've been on this call are going to be part of in the future, because we need your voices as well. And then we go back to the patients and we say, here's what the doctor said, which parts are good, right? Because at the end of the day, the experts are the Black patients. And we want to hold some teach back sessions in order to get to our community partners and our Black patients who are going to help guide the future of this work. So it's a really big challenge, but if you want to join us in any way, please just reach out to me. I'll put the link in the chat. Thanks.
So fantastic. Thank you so much, Amanda. I said we're going to need some teach back to the medical professionals and trainees as well when you have the results of this. I can totally imagine this. I have a lot of ideas. So this is wonderful. We're running a little bit late, so I want to make sure we keep going. And Kimberly is going to provide us a update on the Illinois Gynecological Cancer Commission. And please, we're going to then be doing an evaluation, and I don't want to lose everybody's feedback. Very, very critical for us to be able to improve and do things in the future. So hit it off, Kimberly.
Yes, I'm going to be brief. So we do want to spend that time on the evaluation piece. What I want to say is when we began this in February of 2020, in our first in-person, I asked everybody to sort of do this phone tree call to their representatives to tell them about this commission. And yes, it took almost a full year after writing the legislative language and asking my representative to in the House to present it to the General Assembly at the state of Illinois. At least 1 year later, it was the resolution was turned into typically or figuratively law in terms of creating an Illinois state special gynecological commission. And Camille Lilly, who couldn't be with us today, but has been instrumental in pushing it from just being a commission on paper to actually now getting those appointments put in place. And so I'm so incredibly touched by how this has moved from 2020 to 2022. We're looking forward to 2023 actually being a group that is going to focus on gynecological cancer in the state of Illinois. But most importantly, how well is the funding distributed, right, for diagnosis, treatment, and survivorship around the gynecological cancers? Because we need a little bit more of that money, specifically because so many of our cancers are unscreenable. And so this is the type of work, guys, as a patient advocate, you can do. So if we do have some folks that are not necessarily from the state of Illinois, if you are interested in the language that would help you create a commission in your state, just put your email information in the chat for me so that we can spread this nationwide, that every state has a gynecological commission. And so having said that, we will move directly into our summit evaluations.
We wanted to share the summit evaluation. Please use your phone link. There's a link there. We can put the link in the chat as well. We will be really looking forward to getting as much response as we can, because this really helps us to make the programming better and show things that you want to see next year. And if there's anybody who wants to throw in that comment section their own email or something else that they want us to contact them, please do. We may be contacting you through the Eventbrite just to say, hey, if you're interested in doing more, like, can we email you? And so we're going to try to keep this community going. I really appreciate that. I did want to show also, if everybody gets a chance to see that, the virtual swag bag. I want to say a very big shout out to some of the behind the scenes team members. Cheska Zaletta is here, and she's probably not even showing her face, but she's one of our incredible research specialists at University of Chicago who really went above and beyond in terms of creating some of the PowerPoint and this virtual swag bag of like fantastic resources, upcoming events, and just links that are direct links. So you can get this via this, the QR code. We'll put the link in there so that you and any of your friends, families, coworkers, anyone else, you can share this virtual swag bag and have a great place for GYN cancer information.
Yes, and shout out to all of the service provider and advocacy organizations that gave us videos and helped us with putting them together in this swag bag.
All right, that's our closeout. But again, please continue to take the time to do the evaluations. We really, really, really need that feedback so we can expand and do more. So we really want to hear from you. And we look forward to starting our planning for next year already.
Indeed. Thank you all for spending the time with us this Saturday. Have a great rest of your day, and make sure that you be assured that everything in the chat we're going to capture, and we will make sure it's a part of the swag bag so that you get all the information that you were looking for, including the recording of the presentation today. Thank you all so very much. Have a great one.